Started again. So I believe that Boris is back with us. Right here. Awesome. Everyone's been asking about you, so we're thrilled that you're back. We want to hear all about your speech. You'll have to let us know how it went. So this is our third and final panel discussion for the day. This one is entitled Existing Systems and Infrastructure for Clinical Networks and Operations for Threat Response. So we have another great set of panelists for this discussion panel. We have Jeff Brown, who's an associate professor at Harvard Pilgrim Health and Care Institute. John Grabenstein, executive director of medical affairs at Merck Vaccines. Elizabeth, or Libby, correct? Higgs? Libby Higgs, who's the Global Health Science Advisor for the Division of Clinical Research at the National Institute of Allergy and Infectious Diseases at NIH. Luann Lance, a public health program nurse, Clinical Operations, Bureau of Communicable Disease Control, New York State Health Department. Amanda Peppercorn, Senior Medical Director, Infectious Disease Research and Development at GSK. And Scott Proestel, I think it's spelled wrong in here, Proestel, Director, Division of Epidemiology, Office of Biostatistics and Epidemiology, Center for Biologics, Evaluation and Research at FDA. So welcome, panelists. So this morning, we had a couple of presentations that were setting definitions for the day. Were y'all here this morning for those presentations? There's one about the clinical network system in the United States and one about ConOps. So just for your information, we sort of level set this morning to go over that. For many MCMs, a public health emergency is likely the only opportunity that we have to collect safety and efficacy data in humans. This means it's very important to leverage the existing infrastructure that we have the best that we can. So now that you've heard a little bit more about the questions and decisions that we've been talking about with relation to safety and efficacy, to harm and benefit, I'm curious to hear about your experiences with doing safety and efficacy research for MCMs during a state of emergency. Have any of you experienced that before? Anyone can answer? Yes. Well, tell me about it. Tell me a story.
Tell you a story. I'm sorry I wasn't here this morning, so you may have covered some of these issues, but certainly I've worked with many colleagues in the room. And the first one, I guess, was the 2009 H1N1. And I thought, had a fairly effective, cohesive, rapid response on the clinical research side. We had an interagency working group. We identified the candidates that needed to be studied, and we worked closely with FDA, CDC, industry, put together BARDA, helped coordinate. We put together EUA enabling protocols. And we even competed at NIAID, an IDIQ contract for clinical research sites. And at the time we were working with Roche and GSK. And it was very interesting that the things that we ran into, even though we had protocols that were done in 2 weeks and thought to be able to produce the safety data that we needed to enable the EUA, we couldn't start them because the sites that we had contracted with, the data access issues were a problem for the companies. And that was a little bit challenge. Now, I don't want to sound like I'm bashing the companies. And with Ebola, with the 2 lead candidates, one of them became a Merck product eventually. The companies worked with us. We were able to put both products into the same study, comparing them not to one another, but to a placebo. And I think that's an excellent model for the future. But I could go on about lessons learned, but those are 2 vignettes just to get us started.
Anyone else have any stories to tell?
Well, so not emergency circumstances, except that it was preparation for war, which was my pre-Merck time with DoD, and getting widespread smallpox vaccination, widespread anthrax vaccination. But the piece lesson learned for this group might be we took our immunization information system and put into it as a contingency ciprofloxacin and a variety of other non-vaccine items in case we needed to, in a hurry, have distributed at every National Guard base in the country, et cetera, the ability to record drug exposures, medication administration, as taking existing information systems and making them hybrid data collection tools.
I've had experience at least designing protocols for animal rule approvals. And I know just from our standpoint, efficacy was very difficult to figure out, well, how would one determine that? What is your comparator? And we didn't think that there would be much appetite for placebo in this type of circumstance. So who are you comparing your hopefully even when you have very well collected data, standardized, if you don't know who to compare it to, it becomes very hard to know if you were successful. And so it's a pretty, it was a pretty disappointing endeavor on my part. But the only two options we could come up with would be individuals who declined the countermeasure for whatever reason, and looking at time to exposure or time to first dose, although that is rife with potential problems as well, because the individuals who present later in the course of their illness or the course of their exposure, even if you know when they were exposed, may be different, and that may be hard to control for. So it was, I was interested in the mentioned placebo, but we really wrestled, at least in these particular animal rule approvals, to convince ourselves and the company that that would be a reasonable path.
One of the things that I would like to stress from the Ebola outbreak and any outbreak is that the rules of clinical science don't change in an emergency. So if we can't determine safety and efficacy with data that's not collected in a systematic way. And it's our job to explain, it's our ethical responsibility to explain to the public that these experimental medical countermeasures have risks. I mean, we've been involved in a study at NIAID where the AIDS vaccine actually increased the acquisition of HIV. So the reason why we do these rigorous studies is to obtain rigorous data. So I do think there's a place for EHR data, but it's not in determining safety and efficacy. And I think a lot of, we tried I shouldn't say we, there were several attempts to do study designs that didn't adapt a rigor in terms of an adequate control. And in retrospect, it looks like some of those may have been harmful to the individuals who had Ebola. So we just have to prepare adequately, we have to lead, and we have to do rigorous science.
You may know that the three African countries had 3 different study designs. So I think Liberia is the one you're mentioning. Guinea, the placebo was time. It was a delayed vaccination. The control arm was delayed vaccination rather than prompt vaccination.
So that's another point that we've learned from Ebola, is that we have to have a U.S. government lead. And there's now the Security Council's put together a playbook for international responses. The ASPR, with Christian DeBoard, who's here, has put together a lessons learned for HHS and identified the NIH as the biomedical research lead for outbreaks and health emergencies. And I think the clarity of who's going to do what across the government, both for preparedness and response, is going to be very helpful.
So from the practice perspective, when you are put a operation for response into action, how is research typically integrated into that? Or is it?
Well, I think, my name's Luann Lance, and I'm with the New York State Department of Health. And just a little background, I spent about 2 decades as a clinical director at a local health department. So I, too, like Rona earlier, am looking at things from a local and now a statewide level. I think research into a product, what we're looking at, my primary role is, activation once it's been authorized, once it's been approved for safe and effective use. That's when we would put something into place. And I think one of the key things that are important for us to know is what appropriate medical screening is necessary for us to safely deliver a product. So we really depend on research and science to be able to tell us what are the potential contraindications, what are the potential risks that we may need to build into an algorithm to really safely either administer a vaccine, a medication. I go back to smallpox, I'm really dating myself now. But there were a lot of risks about heart disease and certain things that we needed to assess to determine whether it could be safely administered. So that kind of research is very, very, very important for us when we activate a medical countermeasure response.
Any other practice-based experiences?
So I'll say that most of my day job is trying to figure out how to use the data that are floating around, mostly administrative claims and EHR data, to help support these kinds of questions. So in a way, I'd say if you can find a date and a person, we can help, right? Because then I can track them. That's the only two real pieces of information we're looking for in that scenario. But often what we're really looking for is how do you use, particularly in an emergency, how to use the data that are already being collected in order to inform. It might not be the answer, but it's, I hope, and this is actually a decision someone has to make, is it better than nothing? So is using some of the EHR data that are there and available better than not knowing anything at that point in time? I guess I've spent, well, I'll probably talk about it a little later, but it will come up, a lot of time building a network for FDA, which is, so the Sentinel system is a network of medical product safety surveillance. On the routine basis, there's not an emergency until the commissioner calls. And then all of a sudden, you need something very quickly. And then you're kind of into, I need an answer in a day, I need an answer in 2, and we can also do that. You try not to do that every day, but we've built a system that can do that in that emergency scenario. It's a lot of work up front in order to be that responsive.
So I'm curious, what are some of the things that are different? So if you're doing clinical research for any drug out there versus when you have an emergency situation and you need to do rapid research to test efficacy and safety, what's the difference between the two types of trials? So
we're working hard, I think, to think through some of those things with our colleagues on regulatory science at FDA and elsewhere. You need efficiency, expediency, you have to be pragmatic, the designs have to be flexible to the availability of the products. Some people have argued for a Bayesian approach. We have a protocol we did with ZMAP that our statisticians called slightly Bayesian. The DSMB has to be very closely involved so that they almost become, they're independent, but a very critical part of the study team so that they can pull the trigger if you begin to harm or help so as soon as that's statistically hit. So we have learned quite a bit from our experiences in recent outbreaks, and we're trying to codify that in generic protocol designs. Also, I think it's critical to be extraordinarily focused on your goals of mitigating mortality, ending the outbreak, and developing rigorous regulatory level data for future outbreaks. And so those 3 goals really should shape what you're doing. There's a lot of other really interesting things, of course, that can be done, but those are the primary goals. And understanding something we did not do well in Ebola was understand the natural history, optimize clinical care, and those are things that I think can be prepared for in adaptive study design. So you want to be you want to be faster, more adaptive, more flexible, but you can't give up the rigor.
I think scale comes into it too. I mean, if you've got 12 botulism patients, it's a different thing than a crop duster just moved across a major metropolitan area. And in which case you may, FDA may well want to know 100 things, which are the 5 most important, because you may need to do a large simple trial instead of an exhaustive, massive case report form.
That's what we basically wrestled with, was not knowing. We knew what the intended use would be, but we didn't know the situation, and so it makes it kind of hard to write that case report form. But one strategy was, say we would only collect serious adverse events. And so I think to the best of your ability, you try to write a protocol and a case report form that is manageable, that's reasonable for the situation, so which can be hard to predict.
What are some of the other challenges that you faced when trying to stand up clinical trials in the midst of an emergency?
I can go ahead. So
IRBs.
I've been involved with a couple of different biothreat programs at GSK. One was an anthrax antitoxin program that was approved by the animal
Can you
rule.
move your mic up a little
Oh,
bit?
sorry. Can you hear me now? No,
Is it on?
maybe not.
On the top, there's a little switch.
Yeah, green light.
Can you hear me now? Okay, great. So, so I've worked with a bunch of people in the room on an anthrax antitoxin program and a pandemic flu program at GSK, and have suffered all of the lack of learnings that we now have. Both programs, really struggled with that control group and with interpreting, even if you get really good data, without a control group, with channeling bias to the sickest patients, with, all of the confounding of these severely ill patients, it gets very hard to interpret the data. So that control group is so key. I forgot what your question was.
Why is it so hard to do clinical research in the midst of an emergency?
Thanks. So because the antibody to the anthrax toxin was approved with the animal rule, that means it's never actually been given to patients with anthrax, and in terms of looking at safety or effectiveness. And so one of the post-approval commitments was to design a field study. So, if and when the antibody is deployed from the SNS to a location for either a local or a massive event, we would be able to be on the ground and collect safety and effectiveness data there. And that is a really difficult study to design, because you don't know the scale of the event, you don't know where it's going to be. The health system will be stressed, so you can't come in and ask, physicians in a critical care situation to be, doing tons of data reporting in real time on an ECRF. So we've really struggled with that. We struggled with a lack of control arm. We proposed using some scavenged blood samples to try to get some real-time PK and antibody assessment, but I don't really have any answers. It was just a real struggle, and we ended up working with a CRO and having to pretty much keep that protocol up and running, for the near future, kind of awaiting that event, which hopefully will never come.
Are there challenges to doing clinical research, clinical trials during emergency situations?
I think there are some lessons learned. I think that it's very helpful that at least on an international scale, we've had government-to-government partnerships, and that's really critical to leveraging the resources that you need, both locally within the host country and within the United States. For example, in Ebola, our NIH director asked for help from other agencies and public health service officers. And we worked collaboratively to get the staff that we needed and to bring in the clinical research expertise that we needed. So we had colleagues from many other different agencies working with us. It's important to work with the emergency operation system and to leverage what's going on and to listen to your colleagues. And when we went to Liberia, they told us SMC is the most critical factor for your success. And I said, what's SMC? I didn't know. It was their social mobilization, community engagement, and communications. Of course, you know, we did a very robust program, and that led to our success. But it's this partnership, and always just keeping and recognizing you can't cut corners, and starting from the beginning and sort of agreeing to the principles, the compliance with international research guidelines, and that's really critical, that partnership, that trust, and agreement on the principles by which you're going to conduct your research.
And it was not just bilateral. It was not U.S. to host government. There were the Norwegians and the Canadians and the variety of other European countries and the African host countries themselves. I mean, our list of Merck's list of acknowledgments of partners is in really fine print because there are so many on it. It was just an incredible experience.
Any other challenges? I'm sure there must be more. Challenges to doing clinical trials in the midst of emergencies.
I certainly think the key is to have it in place and ready before the emergency. I mean, it takes a long time to design a protocol and to get sites. I don't think you would do it. Well, maybe you did do it for Ebola, but in general, it would be far better to think in advance about what the protocol would look like, have it in place, and you could even ship it to various sites. You could have protocols that are already written, in effect, that would be shipped to various sites that would be capable of implementing the protocol if needed. But to do it after the fact is, well.
Implemented, we were asked by FDA and various other public health agencies to start our IV neuraminidase program at the start of the pandemic, and it had been sitting on a shelf. I mean, there had been no work done on it. And so we had to design the study, get the sites, do the contracts, IRBs, ethics committees, start an EIND and global compassionate use program. And even though the pandemic was going on, we didn't get any extra attention from IRBs or anyone really. In fact, there was perhaps more scrutiny and structure, and we made the decision to include pregnant women, critically ill patients, children, knowing that that was the right thing to do, but those vulnerable populations also meant more scrutiny of the protocols, more time to get the sites up and running. So it was a real challenge.
As I mentioned, I mean, that example, we had competed sites, but then it was the company policies, I think it was GSK, I can't recall, that couldn't then use those sites because of their internal policies. So I think having things prepared in advance and agreements, we couldn't work past that impasse. And then you all had to compete your own sites, which delayed it a few months longer. So some things we could expedite. And we wrote a quick natural history protocol at NIAID and put it into one of our clinical research networks, the CASG. And then those sites, they were universities. They were so slow to come on because it went in the IRB queue, and then it took months and months. And the same thing happened with an ASPR protocol that we did for ICU patients with NHLBI. So it was IRB holdup, which made it very difficult.
That seems like part of the...
As opposed to if there had been a single overarching one.
And I think it could be that our new change in OHRP will help us in this regard.
You said the U.S.
In the U.S., yeah.
Our adult cohort enrolled 150 patients in about a year and a half. The pediatric took about 5 years.
That's from start to finish. But what about from idea to go?
Idea to first patient enrolled was about 6 months, which was way ahead of our usual timelines.
For 4 months with our U.S. signed agreement with Liberia to building all the infrastructure and starting the trial. And what we ultimately ended up waiting on was the dose selection.
Because the dose ranging was happening while you were doing the other stuff.
And that's a real problem too. I mean, these studies that you're doing quickly, you're not able to answer the question, how does this fit into the paradigm of care? What combination therapies? how is this drug best given? What are the doses? What are the subpopulations that may benefit the most? It's an all-comers approach, very fast.
And it's certainly, when I used to work at NIH with the HIV network networks, which are global, I mean, it's a big difference between having a site that knows how to implement a protocol and knows GCP and can, basically receive with minimal training a new protocol and implement it versus trying to start up a brand new site. It's like night and day. Mm-hmm. So in an emergency, I think you're going to go with the sites that are already up and running.
And certainly, path of least resistance makes a lot of sense. So let's switch back just for a second to the practice-based side of things. In the event of an outbreak of some sort, or any sort of emergency situation, so be it Zika, RAD, whatever, what infrastructure and systems are available to you, and how do you? What are some of the challenges that you have around data collection?
So in New York State, we kind of have a homegrown system. It's called the Countermeasure Data Management System. And it was originally designed to collect medical countermeasure dispensing data, medical screening information, demographic information, and essentially document the medical countermeasure that was administered or dispensed. Over the course of time, what we found is that there was sort of a learning gap, a training gap. You have this piece of software and local health departments, when there was an event, were dusting it off for an exercise or whatever. We're not proficient or confident in its use. So we kind of went back to the drawing board and said, how can we create a system that is similar in nature for the data collection methods but have separate event types for the same application? So what we did is try to create applications that would maybe fit into more all hazards perspective with either a community reception center assessment, medical countermeasures both with vaccine and pill delivery, and then also a combination of the two, because now we have anthrax with the vaccine and the pills, and try to come up with clinical algorithms and protocols to make the decision making a little bit more streamlined, because in an event like that, local health departments in our jurisdictions, the infrastructure has really shrunk to some extent, and they're depending on a lot of partner agencies to help support them or volunteers. So we needed to try to simplify the decision making as best we could, so individuals that may not have that clinical skill set could still support the operation during mass dispensing. I kind of wrote down, based on a conversation earlier, earlier in the presentations we were talking about response actions, and kind of what we look at is plan for the unexpected with the expected. Use this system or this method of data collection, however you want to define it, in your routine course of business. Every year we have hepatitis A outbreaks. Every year we have seasonal influenza. It's a great opportunity to sort of use this data collection system. It allows us to identify gaps that may need to be re-looked at or rethought. And it also develops a certain level of proficiency with the user, so if something bad happens, there is sort of a baseline of use. And I think that public health, we're sort of guilty of developing a new thing for every response. And when you drill down to the locals, we're siloed, states are siloed, and locals, their silos are really close together, and sometimes they're the same person. And so, to have all these different applications and to drill it down to the local level, it really needs to be something that they're feeling comfortable and confident to use. I will just give an example of how it worked really well. We had a very small jurisdiction that had a very major hepatitis A food handler exposure about 1.5 years ago. It was at a McDonald's, drive-through. And it was near a major outlet shopping area. So there were several thousand people potentially exposed. And we had a secondary exposure with someone with a relationship with the index case who also worked at a pizza place. So, who served wings and celery to the pod workers. This was sort of the never-ending hepatitis A response, right? And there were a lot of nuances, and I won't even get into those as far as social issues, and there was a lot of reason for individuals not being really forthcoming with their history. So we'll just let it go at that. But nonetheless, this little tiny county ended up with between 3,500 and 4,000 doses of either Hep A vaccine or immune globulin, which literally brought the county to its knees. Because we have the capability in our system to seamlessly transition any vaccine-related response into our immunization registry without duplicate data entry, we found that very helpful because folks may present and have already had 2 doses of hepatitis A vaccine and wouldn't require an additional intervention. So we actually had nurses with access to the immunization information system reviewing that upon registration to determine whether or not they really needed to be there or they were adequately protected with the hepatitis A vaccine. So I think that whole integration sort of helped. And we were also able to follow up with individuals that may have wished to have a second vaccination requirement. So I think based on some of those lessons learned, it gives us an opportunity to sort of continue to use a product over the course of time or a software system, whatever you want to call it. So if there is a disaster response, we sort of have one foot ahead of the other. I'm not saying it's perfect. I'm not saying that folks uniformly embrace technology. It's been a process trying to get users to feel confident and comfortable with technology. I'm a nurse, and I always say, it's very hard to pry paper out of a nurse's cold, dead hands. And it's true, because there's that essential distrust of what if the system should fail, and that confidence that we always have backup documentation, particularly with patient-specific data.
Thank you. Jeff, it sounds like in your work you do a lot of trying to see things from multiple perspectives, so trying to bridge gaps between what different users might want in terms of with data. As you've been listening to the conversation today, what are some of the challenges and opportunities that you're seeing for improving clinical trial research, but also integrating research into operations at the ground level?
Sure, it's actually been a really interesting day. It's not what I spend most of my time on, so I'm going to go most recent first. Earlier today I was thinking, the pharmacy systems are actually incredibly good in this country, meaning the pharmacy data, it's live. It is, when you get a dispensing, it's in a system that's 10 seconds later. It is there with your name and what happened. We're seeing flu vaccinations much more through pharmacies now. So you push flu vaccinations more through pharmacy, you have a live system of flu vaccines in the country versus them being in all the other kinds of places you can get that. So that's that's an example of an existing system. It is set up to know who you are and what you died, right? It knows the date it happened. And it's already there. And there's a pharmacy in basically every town. So do we need another system, or do we try to leverage that? If you have to give mass vaccination or some mass dispensing, is there a way to actually leverage that? It's got an ID, a name, and all of those things. So that's just something that I happen to be thinking through during the day. Who's the user? So who's the audience? What do you really need, and how precise do you need to be? How quickly do you need the information? So this is really how we started building all of the networks that I've been part of building. Who's the audience? What questions do you need to be able to answer? How quickly do you need those answers? That will dictate the kinds of information you can get. If you need the rigor of a clinical trial, you're talking months and years. If you don't need that rigor and you're willing to accept, I wonder if there's an increase, a change in the trend. I think Adam was mentioning before, I don't know if Adam was here. We're already collecting the data that can show a change in trend. So it's matching the question to the data to the method and the timing. And that's really what I'm always trying to do. So you have to keep asking those questions and try to get out of the circle that we were talking about before. You really do have to stop, ask the question, and then match it. Sometimes the answer is it's going to take a year. because an IRB is going to take 6 months, and unless you want to, you can, that's a different problem. But we do have ways of getting information quickly. Is that going to solve it? It might not solve it all, but it might be better than nothing.
Right, and particularly if you're not, don't have a lot of concerns about characterizing all the baseline characteristics of the individuals. If you're looking for a subtle effect that could be impacted by, does the patient have diabetes or all these other things, sometimes the epi data is less than entirely useful, and there could be a lot of missing information. But if you're looking for a big effect, well, you can quickly find some answers that way.
Look, your trick, now that this is going to happen to some, it was part of the conversation earlier, the trick is to understand how to use those data appropriately. So there was the mention of, if they don't come back, they're fine. Or they're dead. Or they're dead. Right, so that's
the first thing I thought.
Or they're sick in a different health system. Right, so we have such a fragmented system. So, that's the use of the data. What does that missingness really mean? And it might mean they're okay in that construct of that system, but you might not be able to make that assertion.
And is it a rare event or a common event? I mean, the rare event, death might be missed. A common event, death won't be.
So one thing that stood out to me so far, for going back to your 3 questions, the audience, the answers that you're seeking, and the timeliness of those answers, it seems like we have a lot of different audiences that we're trying to get information for, and that we don't always know what answers they're looking for. And the timeliness piece, it's usually like now, like yesterday. We need it yesterday. So I'm actually I'm looking at the FDA folks, because I'm curious to know what you would say to answer those 3 questions. What's the audience? What are the answers that they're seeking, and the timeliness of those answers? Is that something that y'all know? Think on that. We're going to give the panelists here a round of applause. I'm going to send you back to your seats. Thank you so much.